Good evening, everyone. We are here at the beautiful Ocean Club enjoying the sunset. For all of you viewers at home, thank you for tuning in. This night's broadcast and every presentation broadcast is brought to you by Carib Trans. So big round of applause for Carib Trans. We also like to give a big round of applause for Satel, who's been going around the island making sure we have internet at each location. So thank you, Satel. We also like to give a big round of applause for CCG, which is the former Prince Bernhard Culture Funds, and public entity SEBA. And a big thank you to all of our sponsors. We have close to 60 sponsors now. And several of you are at the event, including Ocean Club, Flamboyant Cottage, Marshland Investments, Marie de Seba. So thank you, everyone. And Boomerang. Yes, thank you, Boomerang. And we'd like to thank Juliana's Hotel for sponsoring Eric's stay this week. And our raffle, as many of you know, we have a month-long raffle. You can buy tickets for us at any of our presentations or any day between like 8 to 4 at the tent. So buy raffle tickets. We have lots of great prizes ranging from trips to diving with C. Seba to lots of art product. A big present? You do not have to be present to win. The drawing is closing night on October 27th. You do not need to be present to win, and you can buy if you are living abroad. You just need to send us an email, and we will set that up. And we have some upcoming events. Tomorrow, you can dive with Eric. We're going to be diving at a FAD, so that'll be really cool. And then Saturday evening is our next presentation with insect experts Jan Miringa and Aglaya Buma, and it will be at M&A, which is the former Topos. For those of you who know what that is. It will be at M&A. They're rebranding themselves. And if you attend any of our field projects, such as Eric's Dive, you get a free sun shirt. They're really nice, they're really soft, they fit well, so you should definitely do a field project with us, made from recycled plastic. Eric Schneider is the Assistant Director of Research and Innovation at the Cape Eleuthera Institute. With 10 years of experience in South Eleuthera, the Bahamas, Eric focuses on sustainable fisheries and marine biodiversity conservation. He has collaborated with fishers on projects involving species like stone crabs, pelagic sportfish, and bonefish. Eric also coordinates the Island School Research class, has been teaching marine science since 2013, and is a member of the IUCN Shark Specialist Group. Tonight, he will be presenting on Fish Aggregating Devices, aka FADs. So put up your hands and welcoming Eric.
Thanks. Thank you. Can everybody hear me all right? All right, great. Let me find the clicker. Okay, so thank you for having me. I want to say a huge thanks to the Sea and Lorne folks. It's been great. It took me a little while to get here. Coming from the island I live on in the Bahamas is probably as difficult as getting from, Miami to here, but well worth it. And yeah, I'm Eric. I am the Assistant Director at the Cape Eleuthera Institute, which is the research branch of the Island School, an organization in South Eleuthera in the Bahamas. We have a bunch of different schools and this research department that are a part of this big organization. So I do fisheries biology predominantly and a lot of teaching. So this morning it was great being down just over here on the dock with a couple of the student groups this morning, talking about FADs, talking about fish. A lot of my work recently has been on the stone crab fishery in the Bahamas. I don't think there are stone crabs here in Ceiba. Not quite the right habitat, but it's a big fishery throughout the Caribbean. And prior to that, I spent a handful of years working on pelagic or offshore fisheries. And so tonight I'm going to be talking a little bit about fish aggregating devices, what they are, how they fit into fishing and conservation. And yeah, happy to take questions throughout or at the end. So thanks again for being here. So the ocean covers about 70% of our Earth, which is very evident on an island like Ceiba. And about 93% of the ocean is considered deep sea, which is classified as anything deeper than 200 meters. The world's ocean on average is over 4,000 meters deep, 4 kilometers deep on average. And the deepest point, Challenger Deep in the Pacific in the Marianas Trench, is about 11,000 meters. One interesting fact that I figured out about the Marianas Trench recently, when it was first sounded, so when they measured it, was in 1875 off the HMS Challenger, doing a big oceanographic expedition. When soundings were done by dropping a weighted rope over the side of the boat, waiting till it hit the bottom, measuring the rope. And so that 11,000 meter deep, or roughly 7 mile deep trench in the ocean was measured as part of the 181 miles of hemp rope that were on the ship at the time when they were doing this sounding expedition. So I thought that was really cool. So mahi-mahi here are a very common species in the open ocean. And one unique feature about the open ocean habitat is that once you get offshore, once you get into the deeper water, most of the species that are up near the surface in the open ocean don't ever interact with the bottom, and therefore don't interact with almost any fixed structures. So they're not coming into contact with coral reefs or with rocks or seamounts, sometimes seamounts if they come up high enough. But generally speaking, the only structure that they encounter is non-physical structure, which if there's any oceanographers in here, you might fight me on calling it non-physical, but things like eddies and thermoclines and fronts and things like that. But then also physical structure that floats in the ocean. So as opposed to things on the bottom. So this photo here, sargassum seaweed is a really cool species, or there's a couple of different species. This one is one of the ones that can live its entire life cycle just floating out in the open ocean. It doesn't ever have to be attached to the bottom. And a lot of fish, that type of structure is going to be the only structure they interact with over their entire life cycle. So what is a fish aggregating device? Hopefully the name gives you a hint there. So a device, which is a pretty broad term, that aggregates fish. So that can be natural structure, things like tree branches or palm fronds or mats of sargassum seaweed are considered fish aggregating devices. And I'll start using this acronym FAD. But also artificial objects. So since humans have started putting things into the ocean, buckets and barrels and trash that floats out are also serving as these aggregation devices. And there's this unique behavior that a lot of animals, not just fish, especially ones that live in the open ocean, exhibit, where they're attracted to this floating structure. And this fact has been exploited by fishers, and there's a bit of a fisheries term there, exploit, not in the negative connotation, basically used by fishers for centuries, so for hundreds of years. And the technology and the fishing that use FADs has really increased, especially over the last 50 to 70 years. So here's a couple of examples of what some modern FADs look like, and then we'll kind of go into how they're used and why things are actually attracted to them. So just a few photos from where I live in the Bahamas. The photo on the right, this is a big old naval buoy that's in the tongue of the ocean, which is in the central Bahamas. This buoy that you see here on the right is probably about 5 or 6 meters across and 4 or 5 meters high, so a big old buoy that's anchored in maybe 1,500 meters of water. Really cool photo that my friend and colleague took here. A lot of action here. You see the mahi. You can count a number of juvenile silky sharks there. You have some jacks and some triggerfish. Behind us, there are a couple tuna and a wahoo swimming around. So just a huge variety of open ocean fish that use these structures. This one's a little tougher to see, but there is a floating structure up here on the surface. We're kind of looking up at another FAD that is this right here. So a lot of times, FADs are just constructed with recycled material. anything, I've learned this as a marine scientist, anything you put into the ocean you can't expect to get back. Same goes for fishing. So if you can wrap a bunch of plastic bottles up in an old fishing net, that's perfectly good and a lot cheaper than buying a brand new buoy. And then a lot of fads are fitted with palm fronds, tarps, old fishing nets, just to add a bit of structure underneath the flotation component. So how do fads fit into global fisheries? And there's a couple of different types of fads and contexts in which they're used. So we'll start with kind of the biggest scale in commercial fisheries. So the biggest application for fads in commercial fisheries is in the tuna purse seine industry. So a purse seine is a type of a net that basically is used to wrap around an entire school of fish. Basically a really big drag net that you can either drag through the water or deploy from a boat as you're driving in a circle. So that's the photo here is from inside a big seine net with a school of tuna there. And so in the commercial tropical tuna fishery, so this is, think most things that would end up in a can of tuna. About 100,000 fads are deployed into the world's oceans every year to support the tropical tuna fisheries. And those fads account for just about half of all the tuna that's caught on the planet, which is pretty wild. I had no idea that scale before I kind of started working with fads. So that's pretty interesting. Chances are if you've ever eaten canned tuna, you've eaten tuna that's captured using a fad. And then on sort of different scales, so in subsistence fisheries, subsistence being, you're catching fish either for yourself or for your family or for your sort of immediate community. Fads are used, the commercial fads that are used, those are drifting fads actually. So they're not anchored to the bottom. It's still a buoy and some structure underneath it, but they're just left to drift out because those are the big boats that go out for weeks at a time and can fish out in the middle of the oceans. Subsistence, so slightly smaller scale, typically a bit closer to shore. These are typically anchored fads, and they can be used to target both pelagic or reef fish. So again, pelagic just meaning open ocean, so things like mahi-mahi and wahoo, as well as reef fish. If you put a fad near shore around coral reefs, you're typically going to catch things like snapper and jacks. Yeah?
For the commercial fad, who pays for those?
The companies that, so in most cases, the companies that are deploying those fads are buying them. Yeah.
So they have a conglomerate or something, they all pitch in?
So, it's probably going to be a company that owns a few boats in a certain area. Well, it depends. Some of these companies are huge. But yeah, typically that company is buying the fads that they're using. Now that is, we can talk about that later, kind of a, can be a tricky situation that we discussed earlier is, once you put something into the ocean, in certain places there is legislation to say, this is my trap, you can't touch it. Fads are kind of in that middle ground where it's just a piece of floating stuff that what's preventing somebody else from using it. But yeah, typically the company or the boat that is intending on fishing it will put it in. And we'll loop back to those drifting fads and some of the nuances around those in a few minutes. So yeah, you can have subsistence fisheries that are supported by fads that are typically going to be anchored, and then also recreational fisheries. There's actually a couple of governments, I think, let's see, maybe Puerto Rico, DNR or D something, I believe paid for a few fads, and I'm getting a nod, so I think that's right. And there's been a couple other governments around the world who will pay to fund sort of more robust fads, like that photo on the right that I showed you earlier. Typically slightly more durable materials. If they're paying for something, they're going to make sure it lasts, specifically to support sport fishing activities. So that's kind of another part of fad use as well. So to get into what I think is one of the really interesting parts, why are fish and other animals, but we're going to focus on fish tonight, why are fish attracted to fads and attracted to structure in general? So if you put yourself in the shoes of, let's say, one of these little pilot fish here, or a little jack, there's a lot of very small fish that live out in the open ocean. So one of the first things that comes to mind, if you are tiny and you live in an area where you can't hide, you want some shelter. So if you find a big raft of sargassum seaweed or a log or a bucket, you're probably going there for shelter. And then conversely, if you're a little silky shark or a mahi-mahi, you're probably going to know that that's where the little fish hang out, so you're going to go there for food. So those are the two pretty straightforward ones. This video here is an example of both of those. So this is on one of our research fads in the Bahamas. This is a wahoo swimming up to a school of, I believe, small horse-eye jacks that you'll see in a second. And the thing that it picks up right there is the second half of one of those horseye jacks that about 10 seconds earlier, that wahoo zoomed right behind my back and literally just whacked this fish in half. So that's showing both a nice little cloud of bait fish around one of our fads and then one of the predatory fish coming in to eat it. And then these are the next two sort of hypotheses as to why fish aggregate around these structures that I think are really cool. So the first one is called the indicator log hypothesis. So this theory is based on, so before humans started putting junk into the ocean, the highest concentration of floating objects would have been in two places. So one, outside of the mouth of really big rivers. So rivers export, tree branches and logs and palm fronds from shore, spit them out into the ocean. They also export a lot of nutrients, which leads to a very productive area. So this theory says that migratory fish, and even take mahi for example, the mahi that are in Saba could have come here from 1,000 miles away. They do these big annual migrations. So this theory says that migratory fish that move far distances across oceans would have learned over the course of their evolutionary history to associate areas with a high concentration of floating objects with productive feeding grounds, which they typically were, right? A lot of nutrients, a lot of plankton, a lot of bait fish, and then after that, a lot of predators. The other area where this structure would have accumulated is at the convergence of currents, so fronts or eddies or gyres. So these big circular currents out in the middle of the oceans that basically act, if you think of it like a drain, they kind of entrap and entrain all of the debris in that area. But they also entrain all the prey fish and the little plankton. So same thing, migratory fish would have learned to associate these areas with a lot of floating objects with productive feeding grounds. So that's one. And then the last one I'm going to talk about is the meeting point hypothesis. Again, a little more straightforward, where if, say, for instance, tuna, they form these really big schools. They do a lot of their foraging or their feeding at night. Those schools often will get separated and sort of break up. And then they want to go find their schoolmates again. So if they have a fixed point, like this fad, it's going to increase their chances of finding the other tuna around. And there's been some evidence to show this. There was an experiment, I believe it was in the Pacific, where they basically did surveys on a number of fads and counted the number of individuals in the tuna schools as they were arriving to the fads and then as they were leaving. And the numbers were always smaller as they were arriving to the fads, again indicating they're coming in smaller groups, forming these big schools, and then leaving to go forage again or do whatever they need to do. So the reality is, the reason that a given fish or a given species is at a fad or at a log or a sargassum mat out there, it's probably a combination of these. It's likely not just one, but these are some of the reasons why animals would like to hang out around structure. The other one, that, not to kind of anthropomorphize these fish, but you're in the open ocean ecosystem is very desert-like in a lot of ways. There's just not a lot of visual breaks. I also happen to think that some things are just inquisitive and just kind of want to see what's up. Maybe there's underlying things to that, but yeah, it's likely a number of these in combination. So what I'm going to do is go through a few different projects that we have run in the Bahamas to try to answer this broad question that I was interested in, is can fads also be used as conservation tools? And one thing that I want to kind of frame this discussion around is that it's not necessarily a conservation or fishing, right? It's not pitting these two activities against each other. A lot of the work that I do currently is with stone crab fishermen in the Bahamas, and I happen to have a lot of firsthand experience and belief that working with fishers is the most effective way to get to some conservation outcome. So I don't want to have this pitted as a conservation or fishing, but the fact that fads are this very powerful fishing tool, they are used in research in some capacity, but I would love it if they were used for research more. So I just think we can kind of improve that side of the equation as well. So the first project that we wanted to do was try to design a fad specifically for research purposes. So I'll go through that in a minute. And then we wanted to kind of get an idea of what happens on a new fad when you put it in the ocean. So what does the colonization process look like? That process of all the fish finding it. Do some things always come first? Do the prey have to come first before the predators? And then what does that community of fish on the fad look like over time? So what does the succession look like? And in doing that, we ran a 2.5-year long weekly camera survey that kind of gave us a little bit of insight on that. And then we also wanted to know, what's the extent of the accumulation of fish or of biomass around one of these things? Because there's a lot of theories, one of which is called the ecological trap hypothesis, So this theory says that if you put fads where there wouldn't normally be fads, right? So we know they, historically, before we started putting things in the water, they would have been at these river mouths and in gyres and things like that. If we start putting fads where it's actually not a very productive feeding ground, are we negatively impacting these migratory fish by providing them false incentives? So do they actually want to go hang out by these fads, but maybe there's not a lot of food there? And there has actually been a little bit of evidence in some places where the body condition index, or basically how skinny or fat the fish are actually a little bit skinnier in a couple examples when they are caught on fads than in the same location, but just not associated with a fad. So all of this is kind of also looking on through the lens of this ecological trap hypothesis. We want to make sure that there's not this big negative impact on marine life by putting fads in. So we kind of, to look at that question, wanted to say, how far out does the fad effect extend? And look at the biomass accumulation over distance. And then the last project that I'll talk about, which I think is my favorite, so I'll save it to the last, is a fish tracking project that we worked on. So the first one was just designing a fad specifically for research. So our goals were to make the thing subsurface, basically so you couldn't fish it unless you had the GPS point. And again, the goal of that was not necessarily to keep this fishing tool exclusive, but mainly we wanted to get a really unaltered look at what the colonization is. Also, if I put an $800 tag on a mahi and it gets caught the next day, that would be very sad. So we kept these things. We wanted to keep them below the surface so they were kind of out of sight, out of mind. We wanted to deploy them in relatively deep water because our interests were in the offshore fish. We wanted to make something that was affordable and practical. We are on a relatively remote island where it's pretty tough to get materials. And if these things were useful to others, we wanted to make sure they could replicate them. And then also that they were safely accessible for us to do cool things with, like put acoustic telemetry receivers or hydrophones. So basically, could we create something that is easy, safe, hidden, and easily able to be outfitted with different scientific instruments? This was a very scary dive that we did when we were deploying these things. That's a 2,000-pound concrete block under us right there, over 600 meters of water. But that was fun. So we did come up with a good design. This is what it looked like from the water. So essentially this little diagram on the right, we had a big concrete block. We had about 600 to 800 meters of 2-inch polypropylene line as our anchor line. And then we had 2 steel buoys at the surface that sat about 10 meters or about 30 feet under the surface. Now that depth we found kept them out of boat strike range, kept them out of any storm surge. You couldn't really see it from a boat unless you were right on top of it, but it was still easily free diveable, so easy to pop a camera on or something like that. We learned some lessons and I think came to a good design at the end. One of the lessons was don't use plastic buoys because something with 18-inch wide jaws bit a hole in one of our first buoys. The more challenge with the polyballs, these rubber buoys, were that they expand and contract. And if you're trying to have something under the surface with anything other than steel cable, that line really slowly retracts or extends, and then the buoy expands, and it's just this whole issue to try to keep it the same depth. So anyway, we published a paper that was kind of a template to create this research fad, and we're pretty happy with it. We got 3 out. One of them, so the average lifespan, I would say, for an anchored fad that's not some big Navy-funded steel thing, is probably about a year. As you can imagine, again, moorings, even for dive moorings, just fly off all the time. We had one that lasted about a year. We had one that lasted 4 years. And then our last one is still in the water about 7 years later, which is pretty great longevity. And we got the projects done that we were trying to do. So we were happy with that. So from there, we wanted to start some actual kind of biological and ecological research. So moving on to the next project is looking at the colonization and succession of a fad. Again, looking through the lens of, can we use these things as conservation tools? And one of the kind of ideas that came up is, could we use fads that are intentionally deployed under the surface so they're not fished to over time monitor offshore fish populations. The open ocean is a really difficult environment to access. The fish are challenging to sample because they're all spread out. So again, an advantage of aggregating them in a location, you can study them more easily. So we wanted to see if we could do something like this and if it might come up with any useful metrics. So like I said, once a week for 2.5 years, we went out, clipped a camera on top of a couple of our fads, did a visual sort of snorkel-based survey, and just counted and roughly sized all the fish that were present. We got a couple nice videos that I'll just show because they're nice and gives you a good idea of what the community is out there. So again, this is about 10 meters down, a little school of, I think these are horse-eyed jacks and a couple of mahi swimming around there. And this is pretty typical for what we found our sort of fad community to look like. Usually there was a pretty reliable little cloud of bait fish, usually some type of jacks, occasionally filefish or triggerfish, hanging right around the structure. And then on less frequent occasions, because they kind of come in and out, some of these migratory fish, mahi or wahoo or sharks and things like that. I think this would make a really nice screensaver also. So if anybody wants it, you can ask me later. And then let's see what this next one. A little dusky shark comes up and bumps into our camera there, and it was really cool. On the first pass, you can see in a little bit right there, it actually has a tag right on the side of its base of its dorsal fin. And we weren't obviously able to read that tag off of this video, but interesting to see that some scientist somewhere had interacted with this shark before. And then this last one, you'll probably be able to recognize this one. It's a little grainy because I had to zoom in, but a nice hammerhead there. And actually, I'm going to replay this because the little fish in this video actually demonstrate a really cool behavior here and concept. So you see these little jacks basically swarm to this hammerhead. They're sitting on the fad structure, and they zoom up there and follow it in this really dense little cloud there. And so large animals can actually also act as fads, as fish aggregating devices. And not just sort of in concept, but in practice. A lot of the purse seine tuna activities, maybe not a lot, but a fair amount, are actually on either whale sets or whale shark sets or dolphin sets, meaning they set their net around a large animal or a pod of dolphins or something that they find first, and then they see a school of tuna underneath that. Now, in some places, you can't do that anymore. In a lot of places, it's very discouraged. But frequently, you get a big whale shark or a big whale or pod of dolphins with a school of tuna underneath it, and these little guys are kind of demonstrating that behavior as well. Okay, so this is just a temperature graph. The numbers are a little bit small there, but so this is over the course of those 2.5 years. The colors are just our two different fads that we were surveying on here. So as you would expect, I mean, we're 24 degrees north, so just into the subtropics, but we still do get some seasons. So temperature is varying from about 24 to 32 degrees Celsius, and you get these pretty predictable peaks that for a lot of the migratory fish that we already knew, ask any fisherman in at least the northern part of the Caribbean, they'll tell you exactly when the mahi are there, exactly when the wahoo are there. But what was lesser known is, and I think really interesting, some of the smaller things that really had never been studied, some of the jacks and the filefish and the triggerfish. And I apologize for the low quality of this, but we got a lot of graphs that looked like this. So these are bar jacks on the top, a fish called a scat, another little bait fish on the bottom. And again, the colors in this case are red was fish that we found on our surveys during the summer months, so the 6 warmest months in the Bahamas. Blue is in the winter. And we saw a lot of this, so it wasn't quite as beautiful as that temperature graph. But a lot of these small fish we were seeing seasonality with as well. Now it's really interesting because a thing like a barjack, it might only get that big, not really known to be a migratory fish, but we also noticed that we were seeing a very specific size of barjacks on our fads. It's always fish about this big. Never adults, never the tiny little ones that you might see up in the sargassum. So we think it might be some kind of interesting intermediate stop between when they're tiny little juveniles and basically drifting around with ocean currents. Maybe they stop out off some fads before they sort of recruit onto the reefs in their larger life stages. Yeah. So the days are across the x-axis. Sorry about that. So this is just from when we deployed the fad to 2.5 years later. And then the y-axis there is average per, I think it says 2 point, wait, what is it? Oh no, those are just, numeric values. So number of fish per survey. But yeah, a lot of these, you see a point up at like 600, 700. When with the small jack species and the bait fish, when they're there, you get a cloud of them, right? There's a big school right around the buoys. But we thought this was interesting because, again, this stuff hasn't been really shown for these sort of lesser interesting species, if you will. But from things like potentially trying to monitor open ocean biodiversity, it's important to not just look at the commercially important or the big flashy species. You want to know what the little stuff is doing too. And some of those might be interesting indicators of wider changes. So I will spare you the other graphs, most of which look like this, especially for the things like the mahi and the wahoo that we generally already knew the seasonality of. And another interesting thing that we found was, to spark notes it, fads colonize quickly. So within 2 to 3 months, they kind of hit their peak diversity and their peak biomass, at least at it didn't change over the next 2 years, so we're assuming it probably didn't change much after that. So things find it pretty quickly. And that was kind of, there was some evidence out there before that things like tuna find these things really fast, fish that move around a lot. We thought it would be a little bit slower with some of the small bait fish that are probably a little more just dependent on where the ocean currents want to drift them. So that was kind of cool. And the application of that would potentially be if you want to use a fad as a survey tool, based on information like this, you could know, all right, if I want to get a good idea of what, if I'm trying to sample it once a year, I shouldn't sample in the first 2 months because everything might not be there yet. But if I deploy a new fad, sample it after 6 months, I can be pretty confident that everything that's going to be there is probably there, and then can sample it, monthly or yearly from there. So in addition to just the number of fish and the diversity of fish that were there, we also wanted to look at that kind of extent of the aggregation effect. So that biomass accumulation, how far out does that run? What does that sort of curve look like? Again, through the lens of making sure that deploying fads isn't having some significant alteration on the community around it. There's some ideas out there that if you put anchored fads in deep water near coral reefs, the predators that are attracted to the fads might displace some reef fish. So we didn't test that directly, but we want to kind of get an idea of the extent. So to do this, we ran an echo sounder and a light trap survey. So an echo sounder is basically just a fancy word for a fish finder. If you've been on a fishing boat, that little screen that's usually looking like that, blue and red with little dots in the middle. It's just sending pings of sound down into the ocean, waiting for that sound to bounce off of something, so either a fish or the bottom, and then coming back, and the energy that it returns to the device, you can kind of interpret, how big is this thing. And then in addition, we ran a light trap survey. So a light trap is just a little sort of cone-shaped or cylindrical-shaped clear funnel tube, basically. I might have a picture on the next slide, with a light in the middle. So this is a very common way to capture larval fish and larval invertebrates, so things like little shrimp and little crabs and little baby fish. You put them out in the ocean, you anchor them. The light actually serves as the bait source, if you will. So it's a good way to get an idea of what the smaller stuff out there is. So we basically took 2 of our fads and ran this star pattern with the echo sounder towed behind the boat. So this is the thing here. It's just on this little glider. And I didn't sleep for a couple months because I was dropping $180,000 of somebody else's money into the water every day. But that was what was generating the data you'll see in a minute. We ran this little star pattern here. And then this is a rough look at what the sort of output looks like, sort of in real time, where this would be the bottom here, and you can kind of see these lighter blobs of different color. That's just basically little blobs of some dense cloud of things, so whether it's a layer of plankton or a layer of fish. Here's a better view of the glider there. So the first thing that we wanted to try to figure out is not just, everything that's in the water, but can we separate it out even a little bit more? So you can do some filtering on this data to show you things like what might be a big fish versus what might be a tiny fish versus you can even get as specific as plankton with these things. Now, it's really difficult unless you spend your entire career doing this to discriminate between species or sizes within a species, but we at least tried to get a rough idea of, what big stuff is out there, so basically fish, what really small stuff is out there, so fish food. So that was kind of the filtering that we did, and just our locations here. This is the island of Eleuthera that we were working around. And this gives you a little idea of kind of what this filtering process looks like. So darker or more pink and more yellow is essentially more biomass. So each output would start looking something like this, where there's a lot of stuff in the water. This is going to show you all the plankton, all the fish. And then you can say, all right, take out all the really, really small stuff, all the sort of background noise, the tiny little marine snow and things like that. And then you start to see a couple of these bands. And one of these bands in particular is a really interesting thing that forms called the deep scattering layer, which we saw throughout our surveys. So you can filter it even a little bit more just to look at sort of the bigger of the small stuff. The deep scattering layer, oftentimes referred to as the largest animal migration on Earth, is this concept where just below the photic zone, so where the light stops penetrating through the ocean, on average across the world's oceans is about 200 meters deep. Just below that during the day, there's this huge community of all these tiny little juvenile fish and little crabs and shrimp and zooplankton that want to sit down in the darkness during the day so they're out of sight of predators. And then as it gets dark, that entire layer of organisms moves up to the surface to feed on phytoplankton, which are the plant version of plankton. They have to stay near the surface because they need to photosynthesize. So that layer of food is always at the surface, but the animal version moves up and down every night. And during the day, they're usually sitting, for us it was about 250 or 300 meters, very clear water in the Bahamas. Other places that might be a little more turbid, they're going to be a little bit higher. So we could actually see that deep scattering layer, which is really cool. And then when we said, all right, only show us the big stuff, only show us the fish, we didn't see that much, turns out. We got a lot of screens that look like that. But that's kind of the filtering process. So the big picture here, and then I'll show just the two sort of chunks separated after this, really looks like this. So this graph here, this is distance from the FAD. So we just combined all of those different spokes of that wagon wheel, if you will. So the FAD is basically right here, and then 1,000 meters away, so a kilometer away. And what we saw was after the first maybe 50 meters or so, the biomass dropped off and almost was flat after that. So that was telling us, all right, there is stuff here, but it's right around the FAD structure. And then if you dig into it a little more and just separate it into those kind of two big categories of fish and either tiny fish or plankton, this is the fish graph here. So again, distance away from the FAD. This is a sort of arbitrary measure called nautical area scattering coefficient, which is a unitless biomass proxy. So don't worry about the actual numbers, just the relative measure here. So right at the FAD, it's 50% higher than basically everything else, which was kind of expected. Honestly, just slightly disappointing to know that we didn't have just heaps of fish out there that we just couldn't see with our camera. But there's not any big tuna fisheries in the Bahamas, and tuna are one of the species that would create sort of a bit more of a bell curve here and be kind of this big blob of biomass that moves around. And then when you filter out and just look at the small stuff, what we saw, at least through the echo sounder, was that stuff doesn't really care about the FAD, which kind of makes sense. If it's planktonic, it's just floating around pretty flat. Now, the cool part about this project is when we wrapped in our light trap survey. So here actually is a photo of the light traps we were using. So we're using two different kinds. The one on the top, again, is just a bunch of clear plexiglass cylinders that are all right up against each other with little cracks in the middle. And then you shove a bunch of glow sticks and flashlights down in the middle. So the little baby fish and shrimp and worms and things can kind of swim in between. And then we strapped another one on the bottom that just had a slightly bigger opening, just an inverted funnel in a piece of PVC, but same thing with light as the bait source. So on this diagram over here, we have our FAD there in the middle. The sort of pattern on the top is where we ran the echo sounder survey, and we wanted to sort of overlap that with our light trapping survey. So we deployed these things right at the FAD, 500 meters away and 1,000 meters away. So we had the same survey coverage. And then we did surface or near the surface where our FAD was, 200 meters, 400 meters, and 600 meters down to kind of get the full water column. So we'll shove this over here and kind of go a little transparent to get this graph coming up here, just to remind you of where it is in relation to FAD, 500 meters away and 1,000 meters away. So we actually found really cool that a lot of these little critters, mainly worms and shrimp, a couple little fish, were actually way more abundant right by the FAD. So what we're looking at here is distance from the FAD on this bottom axis here. So FAD structure is right here. This is depth, so the actual buoys of the FAD would be right here, and the anchor line is moving down. The bottom in this location was about 600 meters. And the circles, this CPUE is just catch per unit effort. So it's just a way to standardize for the time that the traps are out. You basically just take the total number of individuals of anything that you caught in the light traps, divided by the time that the traps were out, which we also tried to keep standardized. So we had a pretty substantial, almost in a couple of the replicates, tenfold increase in the little inverts that we were catching right at the FAD. Now again, that doesn't really match with the echo sounder data that we had that just basically showed it was kind of flat all across. But I think it's kind of cool showing that some of these little inverts might actually be actively sort of moving towards the FAD structure. And I think at least enough evidence for some further investigation. There's a big kind of debate on whether fish go or which fish go to the FADs to actually eat there. Some of them we know they distinctly don't based on gut contents. You never find little fish that are at the FAD in some of those fish's stomachs. Some of them we can see eating at the FADs. But I think this is cool and probably something for us to go down. Kind of gets at the mechanistic part behind why fish are attracted to FADs. Maybe there's a bunch of little critters right at the buoys. I have that. That's the same thing just in table form, which I'm going to skip because it's not as nice as a graph. Okay, so on to the last project that we most recently worked on, which was trying to develop a new fish tracking system for use around FADs. So the current, or at least as of recently, state of the science on fish tracking around FADs, you can do it in a couple different ways. So you can use a satellite tag, which typically is good for tracking long-term migrations of fish, but it's not very specific. You don't get a good resolution on a small scale, so you wouldn't be able to tell, oh, the fish swam 100 yards this way and 100 meters this way. You're going to get, oh, it went to that island and then to this country and then back to this island. So that doesn't really help us when we're trying to do fine scale things like look at how fish interact with a FAD in relation to a big net wrapping around it, for example. So the other technique that's used is acoustic telemetry. So acoustic just meaning sound, and telemetry just meaning basically data transmission. So what you can do is fix I'll skip it and then go back really quick. Oh, there it is. One of these, it's about the size of a water bottle, this little acoustic receiver. It's just a listening device. And if you put a little pinging transmitter on or in a fish, if it's within, 2, 3, 4, 500 meters, depending on the conditions, that receiver will be able to tell you, okay, fish number 12, whatever the ID number for that tag is, was here at this time. And if you pay an extra $400, it'll tell you how deep the fish was because it's got a pressure sensor in it. So we wanted to take that a bit of a step further because there had been a couple projects that were really cool projects that used acoustic telemetry to try to understand fish moving around FADs. And typically what they had done was, let's take this FAD, for example. You can attach an acoustic receiver right onto the FAD. Whatever your species of interest is, let's say it's tuna and silky sharks. Catch a bunch of them, either do a little surgery or an external tag with a transmitter. But all that is going to tell you is presence and absence. So is the fish there or not, right? It's either pinging and it's, yes, it's here. And if you got some money, here's the depth that it's at. But you don't get anything about distance away from the FAD. It's just, yes, it's there. Here's when it's there. Here's the depth it's at. But I thought that distance away was a really important metric, especially in your kind of through the lens of fisheries interacting with FADs. So we wanted to figure out if we could create a 3D sort of tracking system. So in 3 dimensions, track fish around FADs in open water, which has the additional challenge of not being able to put stuff on the bottom. So you can do this type of 3D tracking with one of the ways is called a VPS or a VEMCO positioning system. VEMCO is just one of the companies that makes this gear. So you can set a bunch of these receivers on the sea floor, maybe let's say 100 or 200 meters apart. If you're in an area that's a little bit quiet and those receivers can kind of have overlapping fields of view or fields of hearing, I guess, with fish swimming through, you can use a concept similar to triangulation called hyperbolic positioning to figure out exactly where that fish is. Now, if you don't have a seafloor because you're out in 600 meters and they can't hear that far and it's hard to get down there with a scuba tank or otherwise, then you need to do something else. So that was kind of what we were trying to do. And again, through the lens of trying to potentially come up with some information that could either make suggestions on how to fish fads more effectively to either reduce your bycatch or increase the selectivity of the fishing. So that was kind of the aim here. So the first step was to use a bunch of our students here to reel tags up and down for about 3 straight days because we needed to calibrate all of our receivers. So in the diagram that I'm realizing is a little skinny on the right is kind of what we ended up with. So that same research fad that we had installed a couple of years prior, we came up with this rigging system very similar to how you would run a flagpole, and basically took a couple of these receivers and went down to the fad, connected them to the mooring line. And now the interesting part, and we did this by design, the interesting byproduct of those buoys being subsurface is that you have a taut mooring line. And that fad isn't swaying around like most surface fads were. They're just going to move where the current takes them because there's a little bit of slack. We have a very vertical, taut mooring line that you can't actually run gear up and down. So we basically created this little flagpole line, lowered receivers to fad 100 meters down and 200 meters down, ran a bunch of tests with just dragging tags on a fishing line back and forth. And then on one very flat, beautiful day, one of our students who is a great angler caught this barracuda that was on one of our fads that we had outfitted with the system, did a quick surgery to put a little acoustic transmitter in, released it. It was great. We hopped in the water and saw the thing just shoot right back down to the fad. And so that was what we were working off of. And we wanted to try to see if we could analyze that in a way that's going to give us these additional metrics. And then what you can see, we kind of went from there. So again, this was the kind of prior sort of furthest that you could go, right? One receiver on a drifting fad. So this is typically what the commercial tuna fads would look like. It's actually a big like pancake shaped buoy that's about 6 feet across. Occasionally we get them washed up on the beaches. They're these big gray pancakes. And then you can affix a receiver right underneath it. A lot of them are draped with either fishing nets or palm fronds just to add some structure. So this is what had been used before. There have been a couple projects published on that looking at presence and absence data. We took it the next step to create this rigging system down our fad. Now we also understood though that our subsurface deep water fad was slightly unique, and not everybody's just going to have one of those in their backyard that they can run the same rigging system on. So then we also said, all right, well, we should probably do this on a commercial drifting fad as well. That was a bit easier for half of this project. So half of it was the middle component of that far right diagram, where we basically just hung 3 receivers in a vertical line with a weight on the end under the fad. That replicated this here. And so with receivers in this orientation, that in theory, and we'd see if it worked, that in theory would allow us to do the distance away and the depth calculations. So depth we already knew we could do, but you have to pay extra for the tag. Distance away was not possible yet. And then the third one, to make this a true sort of 3D positioning system, was direction or bearing. Now this one was a little trickier. We designed this sort of, you could think of it as an upside down umbrella that sort of opens up in the water to get 3 of these receivers about 10 meters apart from each other. The challenge is when these receivers are really close together, they're hearing the tag and it's thinking, because they're so close, that it's coming in at the same time. And it's actually the difference in arrival between the receivers that is how it kind of does its calculations. So we had to get 3 receivers out in the open ocean as far apart as we could, but on one structure. We came up with this crazy thing that was really cumbersome to put in the water, but was beautiful once it got in the water, with these extendable window cleaning, fruit picking poles with our receivers on the 3 sides. And then you can see the photo's a little distorted because we were using a GoPro, but you can see the drifting fad there. So there's a line that has these 3 receivers and then the 3 vertical ones. And so again, our end product that we were shooting for was a transportable, what we were calling the VAR, a vertical acoustic array. And the great part so far was that the first component of this did work. So when we had 3 receivers in a vertical line, we were able to calculate distance away and depth. So we came up with data that looks like this. So in a really cool way, you can see this is about a 35-minute snapshot of that barracuda that actually did hang around the fad for about a week while it was tagged. We took the receivers up after about 9 or 10 days just because this was a test. We weren't particularly interested in what barracuda specifically we're doing. We just wanted to see if it worked. So across the horizontal axes here, they're lined up, so you have time. And again, this is just over half an hour. And then for this top one, you have the distance away from the fad or from the array. So if it's 0, that animal is sitting really close to the fad. And as the distance goes up, it's moving further away. And then this panel down here is the depth. So 0 would be it's sitting right at the fad, and as the negative depth increases, that animal is moving deeper. So you can kind of put this really cool mental picture together of what that animal is doing over the course of half an hour. It's moving a little bit further away from the fad as it gets a little deeper, and then it kind of changes its mind, comes back towards the fad, but it's still going deeper, and then it moves away from the fad again as it's continuing to increase its depth. So this was really cool because now we have this distance away metric that we could potentially actually run a project off of to say, hey, maybe in the purse seine fisheries that wrap this big 2-mile long, 300-meter net around a whole school of tuna, but it also catches a bunch of silky sharks in there. And we don't want to catch the silky sharks, we just want the tuna. So could we tag some animals with this array hanging under the fad and say, oh, maybe there's a time of day that the silky sharks, do some deeper foraging or they move a little bit further away. So just as one example, but we think we can now start to apply this component of the array and actually kind of find out some interesting information. The second half, the horizontal component where we were trying to do the bearing or the direction, did not work yet based on basically the close proximity of those 3 receivers and what's called a multipath error. So basically as a tag pings on that fish, it's not only it's sending sound in all directions, right? So it's not only sending sound waves straight to the receiver, but it's sending sound waves up that bounce off the surface and then hit the receivers also. So it kind of confuses the receivers, and because they're really close together, we just couldn't, there was basically an unacceptably high amount of error associated with it, so we couldn't trust it. There has been a software upgrade to those receivers. So we do have plans to test that again because we would love to have this sort of mobile unit that we could put in the water, clip underneath a fad or an aquaculture pen, and be able to actually generate a sort of 3-dimensional track of a fish or a shark or something swimming around the fad. So we're not quite there yet, but we're going to still work towards that direction. The paper describing the vertical component is in review right now, so hopefully that will be out for everybody's eyes soon. And yeah, next steps after that are trying to figure out applications for this thing. We've had a couple of colleagues who have some interest in trying to understand how oceanic white tips move around aquaculture pens. They've anecdotally observed some sort of cyclical towards and away from the pen behavior based on the cycles of feeding of the fish that they're growing inside, and are worried about disease transmission, which is a big issue in open ocean aquaculture. So we think we have some cool leads on what to do with this thing. And hopefully, you guys can keep tuned for updates on that. So that's what I got for you. Again, I wanted to thank See and Learn so much for bringing me out here. We have a whole bunch of people who have been involved in this work over the last probably 5 or 6 years, and a number of funders here. But yeah, thanks so much for listening, and I'm happy to take any questions. Okay, I have to remember to repeat the question for the people online, so if I don't, tell me. Yes?
For the commercial fads, do the fishing companies that put them out ever try to retrieve them, either because they might be able to be reused or for ecological reasons?
Yeah, so actually, and you just remind me of something that I forgot to mention, that is sometimes incentive to retrieve them. So when you put a drifting fad that's not anchored in the water. Oh, sorry. Thank you. That's why I said it. Okay, the question was, do these fishing companies that use the drifting fads ever recover them, either to reuse them or for other reasons? So the first tangent is that those drifting fads that the big tuna boats put out are first off affixed with a tracking beacon, basically, so you can find the things, right? One boat might have hundreds of these things out, and it's all in the middle of the ocean. The companies with a little bit more money behind them will also fit them with an echo sounder buoy. So a little solar-powered fish finder. So they can literally log onto their computers on their boats that, again, go out for weeks or months at a time and say, all right, fad number 47 is at this GPS location, and the echo sounder is showing X amount of biomass under it. So let's go fish that one and not these ones over here. So in one way, that alone could, in a well-managed fishery, be a really sort of good sustainable tool for fishing, right? Because if you're not buzzing all around and trying to make dolphin sets and things, you know where the fish are, you go, you catch your quota. that could be a great thing. Now, a lot of them are not recovered, and a lot of these things that are draped with old fishing net for their extra structure end up wrapping around coral reefs and washing up on beaches. The challenge is that where a lot of these fisheries occur is in the high seas, so in international water where a single country does not have jurisdiction over the laws there. Most of the fishing in the high seas are managed by RFMOs, so regional fisheries management organizations. In the Atlantic, there's one called ICAT, the International Commission for the Conservation of Atlantic Tunas. Now, these rules that they put on for FAD use, for example, and recovery mandates, they're not, it's tricky, right? Because there's not one country that's enforcing them. They're not always binding. I would say they probably would not try to recover them for reuse because they are relatively cheap when you're looking at that scale. But I think some of them are probably starting to recover them from some of these policies that are being put in place saying, you're going to get fined if you throw 400 fads out and don't come back with 400. There's also been a really cool project that has been making and testing, they're calling it biodegradable fads. And the most recent version is called a jelly fad. So it's kind of trying to replicate a big jellyfish. It's this big bamboo structure that has some natural material cloth on the sides of it that slows it down a little bit so it's not going to drift out of the fishing territory as quickly. Will biodegrade. And another big push was to get these companies to not use netting underneath. So there was a crazy paper that was published a number of years ago that estimated that 500,000, so 500,000 juvenile silky sharks died by being entangled in fads every year in the Indian Ocean. I think it was the Indian, maybe the Pacific Ocean. That's just from being entangled in the draped netting underneath, not even caught and brought up on deck or anything. So again, there's a big push to use streamers instead of netting, use natural materials like palm fronds. It's always a trade-off, right? Because you put a palm frond in the water and it starts to break down maybe more quickly than the life of that thing. Most of those fads, at least as I understand it, are not really fished until about 2 months after. The tuna don't show up immediately, so they got to be slightly durable. So there's probably a little bit of recovery. I'll admit that I don't have a ton of experience with that side of things, but they're definitely not all getting recovered, or not ever close to all. Yeah. Yes. Oh, wait, microphone coming your way.
So you maybe asked this question quite a bit with fads. You mentioned, for example, the barjacks that are going to show up as intermediates, not as juveniles, not as adults.
Mm-hmm.
So presumably this is part of their kind of ontogenetic migration. That's where they come. At that time of their life cycle, they want to be under that kind of structure.
Mm-hmm.
Can you speak at all to the production versus attraction?
Yeah, that's a... So should I... okay, yes, I will. So to summarize the question, we were seeing sort of juvenile subadult barjacks turn up to our fads, but not the small little juveniles, not the adults. And then the question was just thoughts on the attraction versus production, which is referencing this idea of do fads and also do artificial reefs, right? Artificial reefs and coral reefs in general, depending on how liberal you want to be with your definition, are also fads, right? If you put a pile of cinder blocks or sink a ship, that's going to attract fish to it. So this question is basically when you put a fad in the water or when you put an artificial reef in the water, are you generating genuinely new habitat that is going to increase the amount of fish in that area, or are you just pulling fish from other areas? I tend to think, with no direct evidence behind this, that fads are probably just concentrating and not really creating new, genuine habitat. Mainly based on the fact that, and the juvenile jacks example is really interesting because a subsurface fad is an incredibly unique thing that a fish would encounter, right? Aside from a seamount, a fish is never going to find a randomly mid-water stationary object. If it happens to be mid-water because some log is slowly sinking, it's going to be floating. So that doesn't really occur naturally. So that's probably a bit of a kind of just weird situation there. But yeah, I'm kind of on the side of we're probably just drawing from other areas. And it's kind of related to this idea that, and a lot of places have tried to use fads. This is sort of drawing a different thing, but drawing people away, taking some pressure off of reef fisheries and putting them onto pelagic fisheries. So not to segue your question there, I hope I kind of answered the first half of it. But I think that's a really interesting idea too, because mahi and wahoo, in my opinion, are some of the best fish you can eat from a sustainability perspective. They're very, well, I won't say we can overexploit anything, but on the scale of, on the spectrum, they're pretty difficult to overexploit because they don't form huge schools like tuna, so they don't make sense to net fish. Whereas tuna might form a school of, you know, 1,000, a couple thousand fish. You scoop it up with a big net with one of these big purse seiners. Mahi, I've never seen more than 20 of them together. Maybe people have seen more. Wahoo are even more, not quite solitary, but they might be in 2s and 3s and 5s. So you can't really net fish them. They grow super fast. I'll butcher the numbers off the top of my head, but mahi do something crazy like a centimeter a week, maybe. And I think they reach reproductive maturity in a year or so, maybe a little more, a little less. They grow super fast. They reproduce super fast. They're difficult to catch in mass quantities. So some of these projects to install pelagic fads to try to say, hey, let's catch some of the mahi instead of the, Nassau grouper critically endangered in the Caribbean. I think that's a really cool idea because a lot of the things that do hang out around fads are a pretty good choice. And also, so with the drifting fads, that's kind of a different ball game, right? It's at an industrial scale where you can catch the entire school. There's often a decent amount of bycatch with that, right? Not only, let's say you're targeting yellowfin tuna. Not only are you typically going to catch a bunch of juveniles of that species, but you might catch a couple silky sharks, you might catch an endangered sea turtle or dolphin. Anchored fads are almost always fished with rod and reel or a hand line or a spear. All of those are really, really selective fishing methods in a really good way, right? And some people that I know do spearfish around fads. I think you're a bit insane because of the sharks that hang out there too. But right, spearfishing is probably the most selective method of fishing. You're only going to shoot what you're trying to shoot. Rod and reel is generally really good too because if you catch something that you don't want, you just cut it off. Some of those animals, don't love being caught and released and might die afterwards. But generally speaking, it's pretty good. So I think that's a really cool kind of aspect of that too, is can we use them to draw humans away from reef fisheries and into some more potentially sustainable ones.
Because you brought it up, the Puerto Rico fads,
Mm-hmm.
that is the idea behind it, is to draw the fishermen off the reef and towards more sustainable fishing.
Great. Yeah, that's really cool. I think, and there's a really good research program there. I don't know any of those folks directly, but have followed them. Yeah, the comment there was just saying that program, the Puerto Rican government installing fads, that was the main intention to get some fishing pressure off of the reefs. Yeah, I think that's really cool and something that we should maybe try in some other places. And there's trade-offs, right? Some places you've got to go further offshore before you get to the pelagics. And, if people have small boats, there's safety issues, but I think it can work in a lot of places. Yeah, thanks for that. Yes?
Do the fads draw in like turtles or any of the mammals? Whales?
Yeah, so I would say, yes, the question was, do fads draw in turtles and whales and things like that? My answer is going to be based off of things that I've heard and not direct experience because any fad that I've ever been around are the ones in the Bahamas. I've never seen marine mammals or sea turtles. But if you read some of these papers and some of these reports, those animals are on the list of bycatch species. So turtles and dolphins and I think pilot whales occasionally do come up in these purse seine nets. It's probably a very different set of mechanisms as to why those animals want to be around a piece of structure like that. And it's definitely not as common as the other types of bycatch like sharks and things. But they are listed on a number of these sort of bycatch lists in the active fad fisheries. Yeah. Yeah. Mm-hmm. Yes.
I have a question. So the subsurface fads, are they deep enough that they're not a navigational hazard?
So the question was, are our subsurface fads deep enough that they're not a navigational hazard? In our area, yes. There's no cruise ship, barge, anything traffic. Where we deploy these things is kind of right up in the northeast corner of the Exuma Sound. where you wouldn't be going across that deep water to get to anywhere other than 15-foot deep water. So any boats that are up there anyway would have issues far beyond hitting a fad. That does kind of remind me of a really interesting thing that we have found with our fads. So they're only 30 feet, about 10 meters below the surface, but the 2.5 years of camera surveys, the 5, 6, 7 years that I've been hopping in occasionally and checking all these things and doing other things, we have never once seen any billfish on our fads. So no marlin, no sailfish, but we know people catch those in this immediate area. So it seems really odd to me that just that 30-foot difference would, and even when we were deploying one of them, we were using these big lift bags to kind of get the buoy set. Lift bags broke off on one of our deployments, and it was overnight, so they washed away. We thought they were lost. Some fishing boat called us 2 or 3 days later and said, "Hey, I think I found your lift bags. We caught 2 white marlin off of them earlier, but here's a GPS point if you want to come get them." So in the exact same area, it was 5 miles out. We know that there's billfish there, but we've never seen them on our fads. So there's a couple reasons why I think that might be happening. First of all, there might be a little difference in the sort of bait fish community that would be sort of willing to make that 30-foot venture down to the fad. We've almost never, I won't say never, but almost never seen flying fish, which are very sort of surface-oriented fish down at our fads. Some of those little, sargassum fish and things like that. 30 feet could be a big and pretty dangerous jump for them. But also a lot of those billfish are feeding down in the scattering layer anyway, so I don't really think it's that. Another interesting thing that I would love to run some experiments on is looking at how different types of fish find the fads. So we spent a lot of time this morning with our students going through different activities around this topic. So fish can see, obviously, fish can smell. There is some evidence that fish can smell the biofouling, so all the barnacles and algae and things that grow on fad structures, and might use that to find them. And they can also hear very well, and sound travels a lot further through water than through air. So one interesting aspect, I kind of alluded to this earlier with our subsurface fads, they don't move at all. There's no surge down there. There's no clanking components or waves splashing against it or chains, shackles moving around. So I haven't thrown a hydrophone in the water yet for this at least. They're probably functionally silent, and there's a decent chance, and actually this is, there's some evidence for this in a project that was done off Australia, that fish hear these fads and that's how they navigate towards them. So maybe, are marlin more reliant on hearing to find fads than mahi? I'm not entirely sure. I think kind of unrelated, I would just love to test, what a silent subsurface fad attracts versus a subsurface one that we put some noise on somehow. But yeah, it's interesting. The subsurface aspect has definitely gotten a little bit of a different community of fish around it.
Thank you for the presentation. I'm quite new to this topic. So I was wondering, just on the scale of it, how much in worldwide fishery is fads used to fish?
Yeah.
And how did you get into fads?
Okay.
Such a specific topic.
It is. So the first question was how much are fads used globally in fisheries? I can answer partially answer that. So in tuna fisheries, I think actually the number I just recently saw dropped a little bit from what I had quoted in there. Up until a few years ago, the estimate was about half of the tuna caught on the entire planet by weight. So that's any species of tuna was caught using fads. I recently saw that it's maybe down to about 40%, but still a really substantial amount in the tuna fisheries. Otherwise, I have no idea, and it's really tough to quantify. So I'll give an example of where I live. In the Bahamas, it's known for having 700 islands and keys. There's probably about 20 that have decent population on them. There's actually not a ton of fad use in the Bahamas. There's a few. But for other fisheries, it's really, really challenging for us or the government to get any good fisheries data. Between my lab and where I live is 5 miles. There's probably 3 different landing sites where people bring catch every day. Our island alone is 100 miles long. So and that is the case for a lot of other island countries that use FADs. Long way to say I have no idea what it is sort of globally, but for tuna specifically, it's a really substantial chunk of it. Yeah. And then the second question was, how did I get into studying FADs? Because it is a very niche topic. That's a good question. I'm trying to think. So it was probably a little sort of interest peaked from, we had done a trip to the big FAD that I showed a photo of that's been in the Bahamas for maybe 40 years, that big naval FAD. We had done a trip over there to tag some silky sharks for a satellite tracking project, not related to how they interact with the FADs. Saw that one, it was really cool. And I don't know, we're in a pretty unique location, as are a lot of these islands, where from our lab, you can drive 2 miles in a boat and already be in 600 meters of water. So I think I was interested in, well, my broad interests are, how humans interact with things in the ocean. I do a lot of fisheries work, also some like shark ecotourism stuff. So it was one of those cool sort of human fisheries interaction things. And I think I saw a lot of potential for sort of technology and engineering to sort of be involved, which I'm really interested in too. So things like developing that tracking system. And it's also, if anybody, pitch for my dive tomorrow. If you haven't ever been snorkeling or diving out in the open ocean, it's really, really cool. It's crazy. It's very different from when you can see the bottom, completely blue, big and small things. I'm really excited that we have some fisherman here who has allowed us access to one of his FADs. It's this fisherman. No, not this fisherman. Okay, well, we can still clap for this fisherman. Yes. So we are able to go dive or snorkel an active FAD tomorrow, which I'm really excited about. Most people who are even into diving and marine biology and things have never been in the water with a mahi, and they look completely different when they don't have a hook in their mouth. It's also just really cool out there. You never know what you're going to see. Yeah.
Two questions.
Yes.
Okay, first of all, birds get hijacked by hurricanes and then they spread around all the islands and whatever.
Mm-hmm.
Do fish get hijacked by floating FADs? Question one.
Yep.
And question two, do submarines become a FAD?
Ooh. Okay. First question is similarly to how, did you say birds get hijacked by hurricanes and move around? Plant species move around too, right? Right. Right. So does that happen with fish and FADs as well, right? So does a drifting FAD move fish to somewhere either new or where it shouldn't be? So there's been a little bit of work on that question in the sort of broad field of the ecological trap. So are we providing false incentives for fish by putting FADs where they wouldn't otherwise be? There's been a little bit of, I believe it was tuna and maybe silky shark tracking work, also done in the Pacific. A lot of the FAD work has been done in the Pacific. That's where a lot of the fisheries that use them occur. The general outcome of those projects was fish stick with them for a little while, but they probably don't massively alter their migration patterns because of them. And admittedly, all of these topics, anything out open ocean, there's not a ton of research on them. But with this, there is a little bit of evidence that it's probably not massively displacing things. They might follow it for a little bit. This other project that I mentioned showed that at least in this one area, the tuna you catch on the FADs are a little bit skinnier. So maybe they're foregoing their normal foraging to try to stay by the FAD or something. But it's probably not having a huge sort of displacement effect. Yeah. And then your second question? Yes. Right. Yeah, that's a great question. I have no idea, but that'd be cool to study, but I don't know if we'll be allowed to. I would imagine if we know that whales and dolphins and whale sharks at least temporarily act as FADs to the point where some of these fishing fleets actually use them. I imagine submarines do too. The one limiting factor might be the depth. I've never been in a Navy before, but I'm assuming submarines are typically pretty deep, and a lot of the species that would hang out around fads are, it's called epipelagic, so top layer of the ocean. So mahi and wahoo and tuna and things, they do go deeper than you might assume. And silky sharks, they can go down to 200-300 meters every night to forage. Silky sharks are down at 600 meters to eat little crabs and things. But if submarines are within that range, maybe. Yeah, for sure. And so, I don't also think there's been a project to see if a deep diving whale aggregates tuna near the surface, if those tuna stay with it. There's some really cool, though, associations that have been shown between pilot whales and oceanic whitetip sharks. This is a open ocean species of shark. They frequently, if you see a pod of pilot whales, will see a couple of oceanic whitetips with it. I think I can't remember this project that was published recently, but it was something where either the whales came up with some squid tentacles hanging out of their mouth and the sharks were picking at pieces of it, or vice versa. So there's some evidence that they do sort of cooperatively forage. But again, we know a lot of these, silky sharks and oceanic whitetips have been tracked. They probably still don't go as deep as the whales do. I can't remember pilot whales. Sperm whales forage around 1,000 meters. The sharks might go down that deep. I don't think the tuna and things would. A lot of fish are limited at depth by temperature. They're cold-blooded. Most fish are generally cold-blooded. So the short times that they do spend deeper in those cooler waters, they're eating. They have to come back up to sort of rewarm themselves. Yeah. Maybe. Yeah. Yeah. If you know anybody who will let us test that, then let me know. That'd be really cool. Any other questions?
All right.
Great.
Big round of applause.
Thanks,
for
everyone.
Eric. If you're interested in snorkeling or diving with Eric tomorrow out on a pad, please let Lynn or I know. We will get you on the list. And we'd also like to thank Ocean Club once again for the appetizers and invite you to stay for dinner. We'll see you on Saturday.
All right. Thanks, everyone.